A protein is just simply a large peptide molecule. It's a biologically active peptide that has at least 40 amino acids present, although most of them have much more than 40 amino acids. Because there are so many amino acids in a protein and each amino acid has many atoms embedded in it, the protein molecule is really, really large and has a really complicated structure. The structure of the protein molecule can be described at 4 different levels, starting with the primary level and also the secondary level. In this video, I'm just going to be focusing on the primary and secondary structures or shapes of a protein molecule. In my next video, I'll talk about the more complicated shapes. The primary structure of a protein is just simply the sequence or the order of amino acids in the protein. So this is very similar to just putting amino acids together to make a peptide. This is the sequence of the amino acids connected together with their peptide bonds or their amide bonds. Pretty straightforward. And many times the primary sequence or primary structure of a protein is just indicated with little spheres, each one representing its own amino acid. So something like this. This is a figure that I took from Khan Academy. The secondary structure of an amino acid is more complicated. The secondary structure arises due to hydrogen bonding between 2 amino acids in the primary structure. The hydrogen bonding takes place between the nitrogen-hydrogen of one amino acid, I'm just going to abbreviate that AA, and a carbon-oxygen bond of a different amino acid. And the distance between the 2 amino acids that are hydrogen bonding together to form the secondary structure creates 2 different possible shapes for the secondary structure. So for example, one option, which we refer to as the alpha helix, arises due to hydrogen bondings taking place between every 4 amino acids. Between every 4th amino acids, not between every 4. Between every 4th amino acid. So if you have a primary structure of amino acids like this, 1, 2, 3, 4, like that, then and the hydrogen bonding is taking place between every 4th amino acid, like this, sort of in this sort of order, this creates a twisted shape to the primary structure. That twisted shape's like coiled, kind of like a spring. We refer to that twisted shape as an alpha helix. So this is a twisted shape. And one thing that is not being represented in this particular figure is that the R groups that are on the alpha carbons of the amino acids in the alpha helix, they are all pointing outward. So they're all sticking out on the outside of this alpha helix, kind of like little spikes. So none of them are sticking to the inside of the coil. All of them are on the outside of the coil. So it's twisted, and we'll also add that R groups on each individual amino acid, they point outwards. Now the other option that we have is when hydrogen bonding occurs between amino acids that are a lot further apart than every 4th amino acid, so quite a ways away. There isn't really a specific distance that's required here. So hydrogen bondings, we say that in this particular shape, the hydrogen bonds exist between strands. Between strands of amino acids. So this could look something like, let's say that we have a primary structure with a whole lot of amino acids in it like this, and none of them are hydrogen bonding with each other, and then it just kind of naturally takes a bend like this, and the hydrogen bonding is going to take place between the amino acids that end up kind of lined up among each other in this sort of order. So it's taking place between these individualized groups or strands that are lying side by side. And that's going to give us a shape that looks like this. So here's the hydrogen bonding that's taking place between those individual strands of amino acids. And this is referred to as a beta pleated sheet because the overall shape of this, once they're all connected together, is kind of flat like a piece of paper, but like a little bit like it's been folded up a little bit. So we describe it as being pleated because of those little folds in it, but sheet because it's like a flat planar thing. And on this figure, again, it's not really doing a great job of showing the directions of the R groups that are on each one of the amino acids here, but these R groups on the beta pleated sheet point up and down, above and below the beta pleated sheet. So our R groups point above and below what we would describe as the sheet. Any particular protein can have either just exclusively alpha helixes or it could have exclusively beta pleated sheets, but most proteins, like what's being represented here, have a combination of portions where the amino acids are in an alpha helix and then other portions where the amino acids are in a beta pleated sheet. Typically, there are multiple areas of alpha helices and multiple areas of beta pleated sheets.